Good day, this is the Professor, CBMD PhD, and today's Thursday, May 18, 2023. It is 4:31 and 18 seconds, AM Eastern Daylight Time, and today's research, human genome. So let's hop to it here. Researcher uses mammal DNA to zoom into the human genome with unprecedented resolution. Date May 12, 2023. Source: Kent School of Medicine. As scientists have precisely identified base pairs of the human genome that remain constant over billions of years of mammalian evolution and which play a crucial role in human disease. The team analyzed the genomes of 240 mammals, including humans, and identified base pairs that were constrained, meaning they remained generally consistent across mammal species over the course of evolution. The most constrained base pairs in mammals were over 7 times more likely to be causal for human disease and complex trait, and over 11 times more likely when researchers looked at the most constrained base pairs in primates alone. Individuals born with mutations on these genes may not have been as successful within their species, or were otherwise not likely to pass down the genetic variation. They were able to identify where gene mutations are not tolerated in evolution, and they demonstrated that these mutations are significant when it comes to disease. The team found that 3.3% of bases in the human genome are significantly constrained, including 57.6% of the coding bases that determine amino acid position, meaning these bases had unusually few variants across species in the dataset. The most constrained base pairs in mammals were over 7 times more likely to be causal for human disease and complex trait, and over 11 times more likely when researchers looked at the most constrained base pairs in primates alone. The researchers collect datasets that are applying advances in DNA sequencing technologies to understand how genomes generate a tremendous wealth of animal diversity. The team findings are a significant step forward. They do not understand 99% of the human genome, so it is fundamental to understand which part has been constrained by evolution and is likely to have an impact on human phenotypes. Their discoveries and methods can become crucial tools for further research. The next step for the team is to repeat the process with a primate-only dataset by restricting the subjects. They hope to focus on functions of DNA that appeared more recently in human evolution. They expect this to be even more useful in determining information on human disease. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.